Having said that, let's talk quickly about Pumas. So I would like to first talk about our team. So Pumas is what it is today fundamentally because of the team that we have together. So we have two academic institutions driving the Pumas efforts. The University of Maryland School of Pharmacy at Baltimore and MIT. And Pumas AI is a company that spun off the University of Maryland, and Julia Computing is a spin-off from MIT and founded by the founders of the Julia language itself. And together, we put together a formidable team of world-class experts to develop Pumas. And as we go along, I'm going to introduce the different folks who are responsible for different aspects of Pumas. So, we started this work back in 2017, where we found a lot of bottlenecks in our approach towards integrating various sources of data, real-world evidence, to perform integrated analytics. It was getting very difficult to get the different data sources together to perform integrated analytics, and we found an opportunity to break the barrier to develop something new that was composable and highly scalable. So that was the idea of what got us to Pumas, where we wanted to analyze big data from electronic health record systems. We wanted it to be real time and fast, and we wanted this to be integrated with different kinds of data sources and different kinds of analytics, like the modern machine and deep learning algorithms. And obviously, we wanted something that was easily accessible, not only on cloud computing infrastructure, but everywhere, so that it can be used for real-time decision support system. And obviously, we had to make a choice of how to develop a tooling that will allow us to select a tool that will allow us to do such kind of develop such a product. And it just at that point, Julia was really coming up and about, and all of you have heard enough about Julia, but it just delivers dramatic improvements in simplicity, speed, and scalability. And it was sort of a no-brainer for us to think about what language to choose to develop the next generation of pharmacometric and pharmacological tooling in the context of quantitative analytics. So Pumas and the Julia ecosystem, in short, are designed to allow pharmacometric workflows and pharmacometric analysis to be innovative. You can scale, integrate, accelerate, and be reproducible. So these are the 5 pillars on which we've designed our ecosystem for pharmacometric workflows, where it easily allows you to innovate, scale, integrate, accelerate, and reproduce. So there's this new field that is coming up about. It's called scientific AI. And one of the, actually the lead developer of Pumas, who is also the developer of the differential equation ecosystem in Julia, Chris Rokakis, put together a fantastic blog on the essential tools of scientific machine learning. And the Pumas is actually designed to work with scientific AI. And what is scientific AI? You can go through these blogs to understand more. And we will showcase that Pumas is actually designed to be a combination of domain models, the models that we deal with on a day-to-day basis, plus it allows easy composition with the modern AI algorithms like machine and deep learning systems. So let me first start by focusing on domain models, and I will showcase what are the different modules that Pumas has to offer, and then I will talk about the integrations that we have with AI that are coming up and about. So as of today, this is our vision. Like, we really want all the different modules that we have on our daily analytical requirements available in one system. And it's not only about having these different tools available in one system, but also it should allow easy integration and crosstalk between the different tools, so that you don't have to switch between 3 different software. You don't have to try to integrate 4 different languages to work on the same software. So the different domain-specific modules that we have include NLME, PBPK, and QSP, optimal design, NCA bioequivalence and IVIVC, and the clinical trial simulation with machine and deep learning integration. So I'm going to go over some of them quickly, as this is not intended to be a completely detailed webinar, but I would like to introduce you to what each module is about and what did we achieve so far, or what are we going to achieve in the near future, and then we can have a discussion on any questions that may arise. So given this, let me give you a brief background on the roadmap of Pumas. So we started this project back in 2017. It was under a different umbrella, which is dissolved right now, but we have it was just a coffee discussion here, and right now we are in October 2019. Our latest release include everything from NLME parallelization to routines for PBPK and QSP. We are very excited about some of the new things that we will be coming up with, which is in the next beta release and later in May, which includes GPU parallelism, expectation maximization algorithms, optimal design, seamless post-processing of your models and analysis, and reproducible workflows. And by midsummer, we plan to have our first 1.0 release that can be that will incorporate a lot of these different features. But it's also important to see what is a vision post 1.0, right? It's not just about having these what we have designed so far, but also what is coming up. Post 1.0, we'll have integration with neural nonlinear mixed effects models. So this is more of the integration with the machine and deep learning systems. End-to-end clinical trial simulator. This is going to be all the way from patient enrollment strategies to trial closeout strategies, reproducible workflows from source to submissions, virtual bioequivalence modules, and fundamentally, because of the way we've Julia itself as a language is designed and which we use, innovation is at its core. The language lends itself to innovation in different aspects, and you will see some of the some examples of this as I show you some results based on our work so far. So I would like to first start with non-compartmental analysis. This is a standard analysis that pretty much everyone has to perform right from the first single ascending dose, multiple ascending dose to perhaps phase 3 trials. We have developed industry standard NCA tooling. We have benchmarked and compared the Pumas NCA against other industry standard tooling in over 13,000 data sets, and we have and 13,000 data sets represent 13,000 different scenarios. It can be anywhere from missing concentrations to BQL values to erroneous increase in concentrations after the last observation. So it just represents a wide variety of data sets. And when we looked at these wide variety of data sets, Pumas matches industry standard tooling. Here we're comparing Pumas NCA with PK NCA, the R package that was developed by Bill Denny, based on which which is what Pumas NCA is based on. And we see that we have 100% agreement with all the different parameters of interest, especially in the context of NCA. Right? We've picked both observed parameters like AUC last and Cmax, where I also derived parameters like AUC infinity pred and half-life lambda z, which rely on the regression and the slopes. And we see 100% agreement across these two. More details of this poster and the different data sets can be accessed at bit.ly /NCA benchmark, where you'll be taken to the poster that provides all this information. We also have the very standard bioequivalence module. We've benchmarked this against the industry standard. We have all supported study designs include all of these listed in the table here. We support everything from standard bioequivalence to narrow therapeutic index drugs to highly variable drugs to there are some of the complex Williams designs and so forth. So bioequivalence is also available, and the way bioequivalence works is it integrates very easily with NCA module, and which integrates further with the Pumas modeling modules per se. One of the most important things I think most of you are really interested in is in the nonlinear mixed effects modeling. And, the nonlinear mixed effects systems, the effort is led by Andreas Nowak and Patrick and Vaibhav from Julia Computing, and they just form a world-class team to put together this nonlinear mixed effect system that I'm going to showcase the details of. So we support most features of NLM. We can do both continuous and discrete data models. We support analytical solutions and differential equation-based models, and we have routines for pre and post processing. In terms of estimation methods, we have everything from the standard maximum likelihood estimations, FO, FOC with interaction, Laplace with interaction. We leverage the Julia ecosystem for the Bayesian and probabilistic programming, so we are able to perform HMC nuts, and expectation maximization algorithms like SAEM and IMPMAP are currently work in progress, and we plan to have them pretty soon. And some of the results were presented at ACOG 10. So in terms of NLME, we have threaded and distributed parallelization of NLME simulations and even estimations. So the user does not really have to do additional work to be able to parallelize your simulations or estimations. We just do it for you guys as long as we detect the architecture. As a user, you have really deep control over the differential equation solvers for really high efficiency. You have access to multiple optimizers and cost functions. And the modeling lends itself to integrate with other tooling like NCA and IVC and bioequivalence easily in the modules, in the modeling module. And I can show you an example of this in the next slide. We're also able to perform mixed analytical PK with numerical PD. So you have a PK-PD system. You can do an analytical solution for your PK and write your pharmacodynamics as a differential equation. We also interface with global optimizers and have pretty much the standard tooling for simulation and estimation diagnostics that are required as post-processing routines to diagnose your model. So in order to do all this, we had to develop a domain-specific language so that it's easy for us to integrate different tooling, and it's easy for the user to be able to write your models and to be able to scale to different systems. So this is a simple screenshot of a simple one-compartment model where here everything inside the at model macro represents a model. The param block is where you write your parameters. This is where you define your random distributions. You specify your equations in the pre-block. And this is where in the dynamics block, you either specify your analytical solutions. In this case, central 1 stands for one central compartment. Similarly, we have central 1, depot 1, all the different standard analytical solutions that you can think of. Or if your model is a differential equation model, you can write your own differential equations like this. The next part is the derived block, where you just, you can derive your outputs from here, and the derived outputs can then be sent to, for example, NCA. So, for example, if this is your IPRED, you send this derived IPRED based on the solution of your differential equations to an NCA macro, and this is able to generate your AUC and half-life and any other NCA metric of your choice. And because Pumas depends on the Julia ecosystem for distributions and statistics, we are able to access many different distributions from the Julia stats ecosystem. Here we're just using normal with the mean and a variance. But if you have different kinds of discrete data, you can use binomial, Poisson, zero-inflated Poisson, negative binomial. You can do t-distributions. So it just goes all over. So you have access to multiple different kinds of distributions from within this. So in addition to that, if we have also a function-based interface, which means you don't have to write it as a macro. If you are if you're good in writing your own functional programming aspects, you can write your own likelihoods also in that, instead of using the standard likelihoods here. This is a quick comparison between maximum likelihood estimates between Pumas and non-mem. We matched the parameter estimates and the standard errors. And on an average, Pumas is about 5 to 10 times faster than non-mem. And this is the case for models that are running greater than 10 seconds. And more tests and validations are being performed as we speak. But I mean, 10 is an arbitrary number. I know it sounds very weird, but what I would like to show you is that on the models that we tested so far, and this is just an example of different kinds of pharmacokinetic models that we tested with a large number of subjects, Pumas is at least 2 times faster than non-mem. And I purposely, ordered these by increasing non-mem runtimes. And you can see that as non-mem runtime increases, Pumas is actually much faster than non-mem. And all this is single-threaded. We have not done any parallelization in Pumas when we did this. And we made sure that all the settings are identical between Pumas and non-mem. And this is for differential equation-based models. And now if we go to some really complex models, and here is an example of a really complex HCV model. Here we evaluated in 30 subjects, where non-mem took about 1,800 seconds. Pumas single-threaded for the same model took about 120 seconds. And when you multi-threaded, it was much, much faster. And going from here to here is like there's no change in syntax. You just say, I want to parallelize my estimation. It's just one keyword argument in your function, and you should be able to do it. So it provides you the flexibility and the opportunity to scale and accelerate your models very easily in Pumas. In addition, we also have the Bayesian estimation. We were able to match parameter estimates between for HMC nuts between when you compare it to non-mem, and these are the Pumas results. And I would like to note that we heavily rely on some of the core underlying features of the probabilistic programming languages in Julia, which are world-class in terms of providing not only the algorithms, but also the post-processing of Bayesian results. And just to note, it's not just the Bayesian programming systems, but the statistics ecosystem in Julia is diverse and bubbly. It's growing really fast, right? So all the way from, we have different forms of reading in data sets and reading out data sets. You have access to multiple different formats. The core, the stats base of Julia is really strong, and it's expanding. In fact, some of our the developers of Pumas, Andreas and Patrick, are core contributors to the ecosystem of the statistics, and we have classical inference and the different Bayesian probabilistic programming tools. So next, I want to talk quickly about the ability of Pumas to handle multi-scale models. We can go anywhere from a few ODEs, the simple PKPD models that we deal with on a daily basis, to many ODEs, highly complex PBPK and QSP kind of models. And the ability to scale is primarily driven by the differential equations.jl the ecosystem of differential equations.jl in Julia. I think it's fair enough to say that the Julia's differential equation.jl ecosystem is probably the best of any differential equation ecosystem that exists out there. And Chris Rackauckas, who is a primary developer and maintainer of this ecosystem, has put together a fantastic comparison of the differential equations compared to other differential equation solvers in different languages. And you can see that it's green all the way down. It just says a lot about how we can leverage the differential equations.jl ecosystem for pharmacometric systems. So where do we primarily use this to gain speed, efficiency, and productivity gain? The first and easy gain is probably in quantitative systems pharmacology, right? Probably those of you who are involved in QSP have seen this paper, have seen the 6-stage workflow, and representing the biology to capturing behaviors and exploring the knowledge gap. It's important that this cycle is not only reproducible, but we are able to go through this in an iterative fashion with high efficiency, and that requires easy scalability, right, and speed. So this is just quick comparison between different tools, MATLAB Symbiology, BMS Toolbox that is on GitHub, and the Pumas plus Julia ecosystem for QSP. I would like to note here that a lot of the QSP systems are driven by the underlying Julia ecosystem, and we have some sugar in Pumas to easily facilitate the concept, the scalability from QSP models, the integration of QSP models to pharmacometrics. So the two things that I do want to point out is our ability to accelerate using multi-CPU and GPU acceleration. This is huge, and I will showcase some results in the next slide, which are quite impressive from any standard as we speak. So this is an exam. This is a project that we collaborated with Pfizer's QSP team in Boston, led by CJ and Ted. And this is just a quick snapshot of some of the gains in speed that we saw when we translated a cardiac MATLAB model to Julia. We got a speed of 26x. It went from 1 day to 40 minutes. When we parallelized the cardiac models, we went 115x, so went from 1 day to 9 minutes. And the parallelization was quite trivial. I mean, literally 4 lines of code allowed us to parallelize this model from a single threaded to, so it took us from 1 day to 9 minutes. We saw some massive improvements, gains in efficiency when we took another model, the Lucid model, and we were about 7 times faster in terms of CPU, but 175 times faster when we moved to GPUs. And there's many other examples that we can talk in detail, but this is just an example. Here we had an optimized C code that was being called from MATLAB, and just by comparing with Julia serial code, we had a 7-time speed up just because of the use of different algorithms from differential equations.jl. Then when we parallelize it, there was linear scaling in the number of cores. We saw even more speed up, and when we put it on GPUs, we saw 175 times acceleration over MATLAB plus C. We saw similar acceleration when we moved to global optimization, almost so from about 16 hours to 1 hour when we compared it to MATLAB for global optimization. So the other place where we would use the differential equations ecosystem is in PBPK. So Pumas in general does not provide any of the organism or drug level properties. We are only providing you the ability, some convenience functions to build standard PBPK models. And the most important thing is we allow core integration of PBPK modules with things like NCA, IVIVC, and bioequivalence. So this will allow us to do things like virtual bioequivalence routines, which are getting to be quite popular in terms of the generic drug development. Lastly, I want to talk about optimal design. Optimal design is still work in progress. We made some initial progress in evaluation of the design for the efficient information matrix. We have more of these coming up in later this year, and hopefully a fully functional optimal design by December 2020. And just wanted to quickly show you some of the result that we saw for optimal design. Here is a comparison of the same HCV model where we evaluated the efficient information matrix, and we saw a 200 times speed up of this complex model compared to the original model that was in Puppet. So I covered the different aspects of the domain-specific natures of Pumas, but I also want to talk about what Pumas is in terms of the composability with AI. So Pumas is, without doubt, the first pharmacometric tool that will use neural differential equations for automated model discovery. We presented a concept of this at ACOP, and just as of three weeks ago or 2 weeks ago, the main Chris Lukakis, who the differential equations.jl developer and also the lead developer of Pumas, has come out with this paper called Universal Differential Equations for Scientific Machine Learning. And a lot of Pumas' neural NLME will be fundamentally be based on this concept. And this is going to be coming up very soon in Pumas, where we will be able to do automated data-driven discovery of models. So you give us a data, and we will be able to define what the right-hand side of the differential equation should be. So we'll be able to come up with what the structure of your model should be. And the same system can also be used for automated data-driven discovery of covariate models. So especially in the context of, you finished your phase 3 trial, you're getting a lot of real-world evidence about your drug from post-marketing data. So you get some real covariates that you probably never thought about, and this is where you can really start using some of these machine learning methods, but integrated into the pharmacometric domain to be able to come up with feature selection. Then lastly, one of the highlights of Pumas will also be the clinical trial simulator. So we have all the fundamental building blocks of a clinical trial simulator, because we have simulation, estimation, optimal design, post-NCA, all the different aspects. And our goal is to provide an end-to-end functionality for a flexible, intuitive, and super fast clinical trial simulation engine, which is highly modular, right? And this is currently work in progress. It will include everything from patient enrollment strategies to trial design choices, trial process options like dropouts, loss to follow-up, adaptive trials, and obviously the most important is interim and final trial closeout statistical testing. So it goes anywhere from trial enrollment to closeout will be available as an integrated system in the clinical trial simulator that we'll be putting out later this year. Obviously, a lot of what we do, it's not easy to work on this together without some really good tooling that will allow users to be able to use some of the to use all the modules, right? So we've partnered extremely closely with Julia Computing, and Julia Computing is our front end, provides all the important building blocks to help have a good user experience. So Julia Academy, Julia Pro, which is the front-end IDE for Pumas, Julia Sure, which provides the enterprise support and indemnity, Julia Team for enterprise governance and collaboration, and Julia Run for deployment and scaling. So Julia Computing, as I said earlier, was founded by the developers of the Julia language itself, and it provides supports to multiple different companies across the world. And Pumas is best suited and works best with Julia Computing products because the scalability for CPUs and GPUs in the cloud computing environment, you don't have to do any additional work. They just we made sure that Pumas is integrated well with these systems, right? So this is Julia Team, enterprise governance and collaboration, and Julia Run, which allows it's a single quick deployment and scaling. It just allows you to send your models to the cloud, have control on the number of CPUs or GPUs if you have cloud computing architecture, and also it works both inside your firewall or if you have servers that are out in the cloud, it works well there too. And lastly, none of this is really possible if you're not able to learn about Julia. It's the number one question is, oh, I'm using other systems. I need to learn about Julia. Julia Academy provides on-site and remote trainings and helps you, train your skills. And Julia is a language of the future, and we will help you through Julia Academy to make sure that you get the best of the best when learning about Julia. So with that, I will show you a few references about all the stuff that I've included in this presentation, and want to close out by saying that the whole goal of the Pumas ecosystem is to allow pharmacometric analysis and workflows to innovate, scale, integrate, accelerate, and be reproducible. These are our 5 principles, and we really want to stick to them and make sure that the user has a fantastic experience and the ability to perform any of these without having to bring together 10 different languages and 10 different tools into one environment. This is truly the next generation of pharmacometrics. And with that, I'd like to close out and open the floor for questions.